Very good morning to everybody. So, still we are with the vanadium, how the metal is responsible for different kinds of reactions in biology that we will see. And interestingly, the vanadium management in the biology is important because we are discussing something where this vanadium in some oxidation state in ionic form is basically absorbed by the living system, and that absorption which is dependent on the environment. So, how the environment is responsible? If the environment is typically the marine organisms for the sea, then we will find how the sea water not only containing vanadium, but also some other important species, other important chemical species which can be managed by this particular center, which can sometimes behave as a very good catalytic center. So, today we will just follow the vanadium enzymes part 3, because we are one after another we are continuing for these different types of enzymes. One important enzyme for that system is the vanadium-based different haloperoxidases. And those we have seen that is a very important reaction, because most of the organic compounds that we all know that there are some halogenated organic compounds have some useful properties. Halogenated organic compounds, and if this particular one, that means how we can halogenate a particular organic basic moiety, that can be seen. So, like that of the metal ion, that means the vanadium. So, this vanadium center can act as a very good catalyst. As we all know that V2O5 is also a very good useful industrial catalyst for the preparation of sulfuric acid by the contact process, where V2O5 is the right catalyst. So, not only manipulating, when we have studied the tungsten, when we have studied the molybdenum systems, now this vanadium system is also not only useful for transferring the oxo center, because in the contact process sulfuric acid preparation we basically go for the conversion of SO2 to SO3. But now one more important thing, one important chemical reactivity pattern can be shown by the vanadium compound is that we can handle some of these halides like bromide, chlorides or iodides for the incorporation this bromine in the organic backbone. So, people have tried for different reactions for the direct incorporation of this bromide ion in some large and complex and sometimes medicinally important molecules. So, this is one such example. If we can have highly substituted one, this carbon is substituted by a tertiary butyl group, this nitrogen is also substituted by tertiary butyl group. That means, this carbon does not have any hydrogen, this nitrogen also does not have any hydrogen for bromination reaction. So, if we want to have some other positions where the available CH bonds are there, and we expect that some kind of bromination selectively on those carbon atoms. So, if we get that particular product, then those molecules like our marine natural products can also be useful medicinally. They can have some pharmaceutical importance and we can use them as drugs or medicines. So, very simple reaction has been tried with this particular substrate with that of our vanadium bromoperoxidase. So, this is the living enzyme. So, the enzyme has been taken in presence of hydrogen peroxide and bromide ion. So, the basic idea behind this thing, that what we have studied last time or the previous class, that we are able to form some bromonium ion by the enzyme, the hydrogen peroxide and the bromide ion. So, if bromonium ion is available over there and is interacting through this particular double bond, this CC double bond between carbon number 2 and carbon number 3. So, if it start interacting with this particular double bond, which is much more reactive compared to the other double bonds which are resonance stabilized within the benzene ring. So, what we expect, that we expect that there should be some bromination reaction on this particular carbon, but instead of that we go for this particular bromoperoxidase. Vanadium containing bromoperoxidase is responsible for typical oxo transfer reaction, what we see basically for the different types of oxygen insertion reactions in the organic backbone. So, like that of our molybdenum compounds or that of our tungsten compounds, what we see in this particular case, same reaction is happening instead of the corresponding intermediate based on the bromonium ion, that this oxygen of this particular water molecule which is there in the reaction medium is getting activated by the bromoperoxidase enzyme, and that particular enzyme is therefore going for the corresponding oxo transfer reaction from the vanadium ion. So, vanadium ion is forming over there, and that vanadium oxygen is getting transferred to this particular carbon center to give you the corresponding oxygen transfer on the substrate molecule. So, what we were expecting, that we should have some kind of bromination reaction on this particular substrate, but that we are not getting. Instead of that, we are getting some carbonyl function on the backbone, on the 5-membered ring on the right hand side of the substrate. So, this can also be encountered if we, instead of using the enzyme, we can use the aqueous bromine solution. How the same substrate behave towards the aqueous bromine solution, how the bromination reaction can take place, whether that particular reaction can selectively go for bromination in some of these carbon atoms, because this nitrogen and this carbon is sterically crowded and substituted nicely. We do not have any hydrogen atom available for substitution by the bromine atom. So, this basically give two major products. One is for the bromination at this particular carbon which is already substituted, and as well as the ring bromination in other one only ring bromination. So, we basically get multiple bromination reactions. So, we do not get any reaction where the selectivity can be seen. So, this particular reaction with aqueous bromine is less selective, and we get multiple products, and the bromination reaction for a particular region cannot be chosen. So, this particular direct reaction with aqueous bromine is not very useful one compared to the corresponding bromination reaction using the bromoperoxidase molecule. So, bromination reaction, what we have seen here is that 1,3-ditertiary-butyl 1-indolinone with aqueous bromine can produce multiple brominated species. What we expect? We still expect that if we can make some modification in the reaction medium, that we can avoid this particular carbonyl group insertion in the molecule, we can expect some amount of bromination within the some other substrate or this type of substrate with some modification within the backbone. So, in the next part, what we will see that a typical system in the marine organisms, where we will see that high levels of vanadium we found in the mushrooms. So, the mushrooms are also can have bromine. So, one such variety and one such category has been identified in Amanita muscania, which contains amavanadin. So, we can have some chelating ligand like that of our siderophore. So, this is basically a siderophore type molecule, siderophore. So, it is basically a siderophore type molecule, where we have seen in the different siderophores that iron center was there and the oxygen based macrocyclic chelating ligands are available, and basically those are hydroxamate based, and those hydroxamate based chelating ligands can bind iron nicely for storing iron for biological requirement. So, hydroxamates are there for binding iron within some macrocyclic chelate ring. Similarly, if we want to bind this vanadium, so what type of ligand we should use to bind vanadium in the biological system? That means we are talking something which we can consider it as the vanadium storage molecules. Vanadium storage system. So, how we can store basically, because we can have some free vanadium ion, and what are the typical ligand system that we basically get for the corresponding vanadium binding. So, if we have this carboxy end bearing dipropionic acid based ligand, which is 2S 2 prime S N hydroxy imino 2 prime dipropionic acid. So, N hydroxy imino, which is very much similar to that of our imino-diacetic acid, because imino-diacetic acid we all know is a very standard ligand system. Which is IDA, imino-diacetic acid. So, basically this particular one is a very useful NO2 type ligand. So, nitrogen donor, 1 nitrogen donor and 2 oxygen donor, NO2 donor ligand, and one such ligand can bind one vanadium through this oxygen, and this nitrogen. So, similar to that, only difference in this particular case is that nitrogen, now we have that NOH group. So, we have this the carboxy end, and on the left also we have the carboxy end. So, what is the difference between this imino-diacetic acid and this N-hydroxy imino? So, N-hydroxy imino, this the difference is only N-hydroxy imino. So, how this particular NOH group compared to the NOH can function towards the binding of the vanadium ion, that we can see. So, if we have this vanadium center for this imino-diacetic acid, so this particular one, so this is the nitrogen one, this is the oxygen, and this is the oxygen. Similarly, on this side we have nitrogen, oxygen, and nitrogen. So, very standard and very regular octahedral coordination can be seen if we use this IDA ligand for vanadium binding, and two such IDA molecule can bind vanadium, and depending on the vanadium oxidation state we can have that overall charge N plus on the molecule, on the typical isolated molecule. So, whether it is cationic, we should provide some anions to crystallize it from the solution. So, when we have the NOH function in it, and how this vanadin can bind in the mushroom to take up vanadium from the molecule, but the reactivity for this what people have studied afterward, because synthetically this particular one can be made in the laboratory, and when we react with VO vanadilyl acetate anion in water, so vanadilyl acetate can in water, giving rise to removal of one water molecule and one acetic acid, giving rise to vanadine. So, in the same way, what we have seen just now, that for imino-diacetic acid, where NOH group is not present, only the NH function is present, and two such molecule can bind to the vanadium center to give this particular type of octahedral compound as vanadine. So, vanadine would be very useful molecule, because these low molecular weight ligands can be available in these mushrooms, and vanadium can be stored nicely over there. So, in this particular form, what we see that this vanadium can form 4 such binding positions like this of the carboxy OH groups. So, 4 such groups are there, and these NO functions can function or bind differently, where when we have the imino function, we direct binding for this nitrogen to the vanadium, this nitrogen to the vanadium, but when N-hydroxy imino function is available, what we find that this vanadium is basically pointing towards the nitrogen oxygen bond. That means it is forming a triangular chelate ring, where both nitrogen and oxygens are bound to the vanadium center, which is very much similar to that of our binding of peroxide anion to the vanadium center. So, this sort of 3-membered ring already we have seen is possible for peroxide binding to the vanadium center in bromoperoxidases, in chloroperoxidases, in other peroxidase molecules. So, here also, since this nitrogen was known that in case of this imino diacetate binding, this nitrogen is there, and we see that this nitrogen is binding to the vanadium molecule, and this sort of binding, if they are not in the facial form, they can be also in the meridional form. So, one face of the octahedron, so vanadium is there. So, basically these two are we can have, and we have 2 of these. So, one such ligands, so this ligand nitrogen and ligand oxygen, and this basically giving the carboxy end of this, and this is another carboxy end of this, giving rise to the binding of this particular half of the molecule. So, this VNO binding, this VNO binding is very much similar to that of our vanadium peroxide binding, what we have seen in case of bromoperoxidases or other same type of synthetic molecules, that this 3-membered ring is possible. So, when binding of this N-hydroxyl ligand is in the same fashion, when we have this tridentate binding like this, and we have the O. So, this particular O giving rise this particular deprotonation, and we have this particular 3-membered ring. So, this also a 3-membered ring for stabilization of this particular ring. So, this binding and these coordinations also are very important, and we basically get the corresponding thing from X-ray structure determination. So, structure of the vanadine molecule can be identified through single crystal X-ray structural determination, and we find that the typical coordination, because if we consider all the binding positions, that means this nitrogen, this oxygen, and 4 oxygen in the plane, and on the other side this nitrogen and this oxygen. So, altogether 8 atoms from 2 such ligands are involved in trapping vanadium in the mushroom molecule. So, what we see that this sort of thing, when it is forming, and mostly vanadium storage is important, and also the electron transfer behavior. So, this vanadine molecule can function as a very good electron transfer molecule, it can show one electron transfer. That means, whatever vanadium center we have in the amovanadine, so is N plus. So, it can donate one electron to the system or any other molecule, or it can accept one electron for any other reducing agent. So, both way you can only go for changing one oxidation state, one step oxidation state. So, that is why we consider it as a one electron redox mediator. So, only it can go for a single electron transfer from the system. And the ligand what is present, then hydroxy dicarboxy ligand is very stable towards hydrolysis. That is why it is not releasing vanadium so easily, because if the ligand is hydrolyzing very easily, only we are ending with some acovanadyl compound, but this particular ligand is not undergoing any kind of hydrolysis reaction. So, they are very stable and we can trap vanadium very nicely within this molecule. And also we can see that if we take the laboratory available molecule like vanadium sulfate instead of vanadium acetate, what has been prepared from the amovanadine molecule has been found to interfere with the siderophore mediated iron transport in bacteria and plant. Just now we were discussing that iron center is there and we can have some siderophore ligand to trap this iron, and this siderophore ligand system is well suited. That means, the cavity size is such that it can trap iron. Similarly, when we can use, if we use vanadium sulfate for the system, and if the same ligand is available for trapping vanadium as well as vanadium ion, then basically this particular one in different bacteria and plants, the availability of this particular salt is only hamper the corresponding iron transport. So, iron transport mediated by siderophore molecules can be disturbed by the presence of vanadium sulfate. Why? Because this particular vanadium ion or the vanadium in non-oxo form can go and nicely substitute the iron bound to the siderophore ligand, and it cannot help or it cannot go for the transfer or transport of iron from one side to the other. So, in both bacteria and plant, this vanadium sulfate interference tells us that vanadium in a similar fashion like that of our iron can be bound to the siderophore type of ligation. And we also see that when vanadium is taken up as vanadium-5 from seawater, so in the pentavalent oxidation state it is initially trapped by the ligand, and inside the cell what we see that when they are getting stabilized, it is not the +5 oxidation state, it is some reduced form of this metal ion, that means the +3 or +4 oxidation state is getting stabilized in aqueous medium. So, how we get that? So, from vanadium-5 we have to get the two other species which are vanadium-4 plus, that means we should go for one electron reduction, or vanadium-3 plus where we have to go for a corresponding 2 electron reduction. So, there should be some available reducing agents which can reduce directly the vanadium available in the seawater in +5 oxidation state. So, this is the native metal ion which is directly available from the seawater by the ligand system. So, you have the ligand system, the siderophore type ligand system, And this particular ligand system, when we all know that when this particular ligand is going to bind the vanadium 5, basically we can manipulate the E0 value, and immediately after binding the reduction potential is going down, and it can be reduced nicely by some biological reducing agent to vanadium 4 plus or vanadium 3 plus. The same thing what we know in the laboratory chemistry, that if cobalt 2 ion is present, that means that any cobaltous salt, cobalt 2 chloride is present, and which we dissolve it in water. When you dissolve it in water, it basically binds 6 water molecules, and these 6 water molecules give a typical coordination environment which has a characteristic E0 value for the oxidation reaction to cobalt 3 plus, and if the magnitude of this E0 value is quite considerable, we cannot get the cobalt 3 compound of the hexaco environment. That means, the hexaco cobalt 3 plus is not so easily made, but if we just simply add some ammonia, aqueous ammonia within the system, which immediately replaces all 6 water molecules by 6 ammonia molecules, and as a result our coordination environment is getting changed from O6 environment to an N6 environment, and now the corresponding E0 value for this particular species drops down, and we easily oxidize this particular compound to cobalt 3 plus, and we know that air is only sufficient for that oxidation. So, the dioxygen molecule present in air can oxidize this particular species, which is not possible to oxidize this one, to give us corresponding hexaamine cobalt 3 compound, which can be isolated as the corresponding chloride salt as a beautiful orange solid. So, the importance of the ligand. So, this is the ligand. So, ligand is playing some important role in manipulating the corresponding E0 values. So, the role of the ligands used to binding the metal ion. So, role of the ligands are therefore important, and in the same way, if we have the ligand system which can reduce the corresponding E0 value for the reduction reaction from the vanadium 5 to vanadium 4 and vanadium 5 to vanadium 3 play, we can easily reduce these two, and we can stabilize this particular oxidation state. The reduced form of the state metal ion can be stabilized in that particular ligand environment, and that can be stored nicely. So, if we consider only the vanadium aqueous ion like that of our cobalt system, that for the reduction reaction for vanadium 5 to vanadium 4 and vanadium 4 to vanadium 3 is not so easy to have it, because in strongly acidic medium at very low pH values, say pH 1 or 2, the redox couples for these corresponding reduction reactions are 1.00 and 0.337 volt versus normal hydrogen electrode. So, these potentials are pretty positive. So, whatever biological reducing agent available to us like NADPH or FADH, so these are not useful to reduce these particular aqueous vanadium ions. So, aqueous vanadium ions like that of our cobalt ions what we have seen just now are not so easily reduced back to a particular reduced form where we can stabilize this particular metal ion in that particular oxidation state. So, when ligand comes into the picture and it binds to the vanadium center, the E0 value is getting reduced and we can use NADPH, nicotinamide adenine dinucleotide, NADPH as the useful biological reducing agent or biological reductant, and in all these tunicates and all other system where vanadium is stored, this particular vanadium center can be reduced to either vanadium 4 or vanadium 3 and they are stored over there very much. So, this particular one as well as this vanadium is also some other other biological systems like the C squares and coulombic cells in the blood cells of these. So, Escherichia C which contains also vanadium like vanadine, but this time the molecule is stored as vanadin. So, this vanadin molecule which is available again from the sea level. So, the C squares which are there can bind and can trap vanadium over there, and these organisms basically are available to concentrate vanadium at a level more than 100 times higher than the surrounding sea water. So, in sea water we have some available concentration of vanadium. So, we can consider the concentration available in sea water is some concentration for vanadium, say vanadium 5 plus, and the ligand is available which binds this particular one and then transfer to this particular marine organisms, and this transfer of these marine organisms for this particular one is also changing the corresponding concentrations within the living organisms, because this particular concentration what is available in sea water is pretty less, but due to this accumulation of this vanadium by the ligand system available there, we can increase the concentration level in all these organisms which is much higher than the available concentration in the sea water. So, sometimes 100 and sometimes more than 100 times concentration or enrichment, vanadium enrichment we can call it, from the sea water can be achieved by all these organisms through nice binding of vanadium by some useful groups. So, in this particular case these are the vanadin proteins, and these vanadin proteins are therefore responsible for binding the vanadium ion, transferring the vanadium ion to the system, and then finally accumulating that particular molecule to the living organism. So, it is this particular protein system which is required for collecting and accumulating this metal ion. So, it is not only binding the metal ion, it in the bound form it basically collects the metal ion and ultimately it is in their system accumulating the metal ion. So, we get higher level of concentration for the vanadium in their body. So, vanadins can be identified in the cytoplasm of such cells. So, if we can like vanadin, we can identify vanadins, and sometimes it can go up to 10 million times higher than the concentration of vanadium what is found in the sea water around them. So, basically it is concentrating within their living system, and we are getting more concentrated vanadium in the living organisms related to these all sea squids. So, if we see that vanadium is thus essential for ascidians and tunicates, and they are stored in a highly acidified vacuoles of certain blood cell types. So, if their blood cells are responsible for generating some vacuoles, and those vacuoles can be termed as corresponding vanadocytes like lymphocytes or any other biological systems which is having some affinity for vanadium ion, we can call them as the typical vanadocytes. So, vacuoles can be formed and those vacuoles can be utilized for binding the vanadium ion and can be storing the corresponding vanadium into the system. So, vanadium-5 which is present in the seawater thus can be reduced, already we have seen in these two oxidation states, and can be stored in the soluble form within the very acidic vacuoles, because these vacuoles are very acidic, and in acidic condition what we see that the electron transfer behavior, already several times I told you that the E0 values for the reduction potential, the standard reduction potentials for these systems. So, we just go for the corresponding electron transfer, whether it is a single electron transfer or a double electron transfer. Also, in some condition where pH plays some important role for the corresponding reduction reaction. So, if the vacuoles, so if the vacuoles, so vacuoles are there and which are highly acidic in nature. So, the corresponding E0 value what is available for this reduction reaction must be well matched to that of our biological reducing agent. So, we find that the corresponding observation that whether your E0 value is less or more in acidic medium compared to the basic medium, that is also responsible for storing the corresponding vanadium in these bodies. So, this is a typical form where in the living site what we see that the cytoplasm is there and vanadium in the highest possible oxidation state, in this particular case vanadium 5 plus in seawater, and is entering through some points in the membrane, and branchial sacs are also there. So, these are basically trapping this particular vanadium, and this vanadium in plus 5 oxidation state, this is the oxidation state, and they are known as metal transporter, and the entire area we call it as the corresponding vanadocyte. And when it is crossing this particular barrier in the cytoplasm, this can be stored in the vanavin, and initially the vanavin, the ligand, the corresponding ligand which is being supplied to binding the vanadium 5 initially binds this vanadium in the pentavalent state, and NADPH is available, and NADPH is there which is reducing this vanadium center from vanadium 5 to vanadium 4. So, for the entrance of this particular cytoplasm barrier, we need one metal transporter. So, sometimes we call this particular regions as the receptor sites. So, this particular region can function as a gate which allows vanadium to enter within the cytoplasm as vanadium 5 plus, and this 5 plus oxidation state is immediately reduced to vanadium 4 plus. So, there will be some difference between the character of the vanadium in plus 5 oxidation state and vanadium in plus 4 oxidation state, as well as their size, their ionic radii is also different. So, in this particular case the gate is allowing vanadium to enter into the system in plus 5 oxidation state, but here for this particular cytoplasm, this is the cytoplasm area, and when it is pushing that particular vanadium within the vacuole, which is highly acidic in nature, we need another metal transporter to transfer vanadium specifically in +4 oxidation state. That means the tetravalent vanadium can only be transferred through this point by a different type of metal transporter, which is different for transferring this vanadium in the pentavalent oxidation state and is going within the vacuole as vanadium 4. And why this particular region, why inside the vacuole is highly acidic, that can also be justified because several ATPases, the phosphatases, the ATP also has some phosphate groups, phosphate ester groups are there, is a triphosphate ester, and that triphosphate ester in ATP molecule can take part in the hydrolysis reaction, and there are some ATPases molecule which are vanadium dependent. So, vanadium can play some important role for the hydrolysis of these ATP molecules, and phosphate groups are being released, and this particular case basically sometimes these protons are utilized for the hydrolysis reaction, which can be considered as the acidic hydrolysis, or sometimes bases are required if the OH function is developed or can be obtained from the water molecule itself. So, which can be considered as the base induced hydrolysis reaction or base induced ATPase reaction, where base is utilized and the vanadium ATPase molecule can release more number of protons to the system. And for charge balancing or the charge neutralization, some amount of anions are also available. So, sulfate anions can be available over there. So, the vanabin in the bound form of vanadium, which is supplying or providing the vanadium in the +4, and sometimes some of the vanadium can be reduced to vanadium in the trivalent state. So, basically what we can consider over here, we have the ligand environment or ligand encapsulation, but still from the charge balance thing or the availability of the anions within the system, we see that the sulfate ions can store this particular vanadium as vanadium sulfate in either +4 oxidation state or +3 oxidation state, and we can go for a corresponding enrichment of the vanadium. That means the vanadium concentration can go up nicely from 32 nanomolar solution, which was vanadium 5 in seawater, and within this particular vacuole it can go up to 350 millimolar of vanadium 3. So, thing is that the system is getting reduced and more number of vanadium ions in the lower oxidation state, either in the vanadium 4 or vanadium 3, can be accumulated at a pH of 1.9. So, you see this is a highly acidic pH where this particular thing is stabilized, and this particular concentration of sulfate anion within the vacuole is in the range of 500 millimolar. So, this particular one is also well matching to that of the vanadium accumulated concentration. It is 500 millimolar and this is 350 millimolar. So, they are well matched, as if the vanabin is transferring the entire vanadium there, and this vanadium is storing there as the corresponding vanadium sulfate, where the vanadium is present in trivalent oxidation state. So, this is basically the entire mechanism for the transfer of vanadium from the seawater within the vacuole system, where vanadium is getting reduced and is stored in a very high concentration. So, in the next particular part, what we will see that how we can have the model compounds, how the model compounds can be prepared to see their corresponding activity, what we see in the biological system. That means the model compounds, how they are taking part in the catalysis reaction. So, one such is the corresponding modeling, where we find that the haloperoxidases, what we have identified, that these haloperoxidases, we have seen earlier that they are very much vanadate dependent. So, if we have this haloperoxidase as the corresponding vanadate is getting trapped within these haloperoxidases, so what we see that the corresponding structural models we can have for vanadium-5 complexes for a particular donor sets. So, if we can get the corresponding model compound or the synthetic analog, so synthetic analog can we have. So, we already know the corresponding vanadium coordination environment, that means what are the donor groups present in vanadium haloperoxidases. So, based on that, basically based on that information, we can choose that 1 nitrogen donor atom and 4 oxygen donor atoms can be sufficient to model a corresponding vanadium center as a environment which can be either a 5 coordinate geometry suitable for square pyramidal or trigonal bipyramidal geometry. So, two geometries we can have. Once we can have a corresponding ligand system based on 1 nitrogen center and 4 oxygen center for square pyramidal, and trigonal bipyramidal geometries. So, situation is such that when we have these vanadium-5 complexes in this particular donor set, and what we have seen in the actual reactivity for this haloperoxidase system, that the corresponding preference for the coordination geometry is for trigonal bipyramidal one, and we have seen that the apical site, if we block this particular apical site by one of the nitrogen atom, that is the histidine nitrogen atom was present in the enzyme molecule, and that histidine nitrogen attaching from the apical side of the trigonal bipyramidal geometry. So, in this particular case, if we choose the ligand system in such a way that the available nitrogen, the single nitrogen for these 5 donor atoms is coming as a corresponding imine nitrogen function, because the imine based ligand systems are known as the corresponding CYP bases, and we know that all CYP bases are very easily we can make from the reaction with that of one amine nitrogen with that of our aldehyde or ketone. So, one imine nitrogen can be dedicated to make the corresponding model compound of the vanadium, and all other 4 positions we can take off by the oxygen functions, because we have seen that surrounding this vanadium, if we can have one nitrogen from the apical side, when the environment is not octahedral, but when the environment is trigonal bipyramidal, this one, the second one. So, once this particular site is blocked by nitrogen, the all other remaining positions what is there. So, all other positions, so they are all based on the oxygen. So, we are happy to make that thing, that we can get this thing like in one ice cream cone basically, and this particular one, because this is not the triangular plane which is occupying the corresponding trigonal plane. So, trigonal plane is like this, and we have this trigonal bipyramidal geometry like this. Vanadium is sitting nicely over there. So, nitrogen is there. So, within this cone, so whatever we can have above this nitrogen, so this all these positions, so they are all our oxygen donors. So, that is the most important thing, and is the more beautiful thing is that we have to do all these things based on only one single nitrogen, and the remaining positions can be all oxygen, because we can have several of these oxygen donors available. They can be from water molecule, they can be from deprotonation of the water molecule as the hydroxide anion, or they can be directly forming bond with the vanadium as the oxo bond, that means they are O2 minus, they are basically O2 minus, and sometimes when in the actual catalytic cycle we have the corresponding binding of the peroxide anion. So, we see that 1, 2, 3, 4 different types of oxygen donor atoms can be available to bind to the vanadium center. So, initially if we go for the making the corresponding synthetic molecule or the model compound based on these, so only we have to concentrate our attention, we have to focus our attention for making the system with one single nitrogen. The rest can be all by the oxygen functions. So, what we get from there, one such example, two such examples basically, that we have these two synthetic molecules we can make. One is basically the corresponding salicylaldehyde, bromo-substituted salicylaldehyde molecule, where you have this is the aldehyde function which is attached to that of our glycine part. This is the NH2 function of the amino acid glycine, which is little bit biogenic therefore in nature, since we are handling amino acid. So, this amino acid with that of our salicylaldehyde molecule giving a very good tridentate NO2 type ligand. So, when it is reacting with vanadium center, it is forming a tridentate capping within this particular plane. This is the meridional plane. So, this NO2 ligands binding this meridional plane, and other three positions are occupied by two water molecules and one oxygen. Similarly, in this other case, in the second case also, if we have a different type of ligand with less substitution on the salicylaldehyde ring, that means it is not bromo substituted one, it is the simple salicylaldehyde ligand, and we have some other groups like that amino acid derivative with alcohol part pendant over here, whether we can have some preference of binding through this oxygen of the carboxy end or the hydroxy group from the alcohol end, that can also be checked. And similarly here also we have again NO2 tridentate binding, one is the oxo binding, one is the water molecule, and instead of this water molecule we have now alkoxide binding. This is R, it can be ethyl or it can be methyl. So, RO minus is directly bound to the vanadium center. So, these two molecules can be synthesized and can also be structurally characterized for looking at the corresponding nature of this NO2 ligand. So, we have this NO and O ligand. So, whether this particular ligand is binding in the meridional fashion or facial fashion, which is very important, because we can have the vanadium site when we fix it as the vanadium oxo bond. So, we can have these three positions which are occupied by these, that means this nitrogen, this oxygen and this oxygen. So, this is the binding. So, we can think of the corresponding occupancy of these positions. So, how the other groups are coming and binding through these positions. So, we can go for synthesis . We can go for the synthesis and we can go for the corresponding structure. So, once we determine the structure, basically why we do the structure? Because we want to know the corresponding bond distances, corresponding angles, and finally, the geometry of the overall molecule. So, the geometry basically tells us that what sort of bond we have if we just consider the corresponding only vanadium water bond. So, the corresponding binding of this ACO ligand, if it is there and which is trans to this OXO function, it can have some trans effect or trans labilizing effect or form the OXO group. That means, it can labilize these water molecules which is trans to this particular water molecule bound to this vanadium and can remove very nicely for binding the peroxide or any other incoming group to this particular site. That means, we basically enhance the reactivity pattern which is trans to these OXO groups of the vanadium center. So, the next day we will see how the different bond distances, the information for the different bond distances can help us to understand the detailed catalytic picture of these vanadium centers. Thank you very much.